Very good afternoon to all the students of BIPC first year. So here we are in discussion with the long answer type of questions, and today is the last session for discussion of long answer type of questions. Okay, and today we are going to discuss the questions from the chapter sexual reproduction in flowering plants. Many a times you got questions from this chapter, most importantly on the types of pollination, and second is about the embryo sac. These two are the key important questions or topics from this chapter. Not just for your IPE examination, the embryo sac structure and pollination, types of pollination, abiotic and biotic pollinators. This is a major concept even for your NEET exam. If you can see your NEET previous year questions, there are n number of questions asked from the ploidy of embryo sac, might be the pollinating agents also. Okay, so very important beta, stay engaged for another 40 minutes and this will be in your hands on how to learn the question, on how to memorize it, and how to have a mnemonic tricks to easily present this question. Most importantly, how you can memorize this so that you can present the answer in a systematic way in your exam. Okay, so first you see the question with a neat labeled diagram. Describe the parts of mature embryo sac, mature angiospermic embryo sac. So we have to describe mature angiospermic embryo sac. Mention the role of synergids as well. So how many times this question has appeared? This question has appeared in the year March 2024, August 2022, 2020 year, again 19, 17, 16 and yeah. In every consecutive year, if you can see, there is a question from the paper, or there is a question on embryo sac. Okay, that important question it is beta. So here if you see, the description of the answer goes like, in angiosperms, female gametophyte is called as what? Embryo sac. Female gametophyte in angiosperm is embryo sac. The most common type is polygonum type of embryo sac, which is also known as monosporic type of embryo sac development. You can also use this term beta. We have monosporic, bisporic and tetrasporic type of embryo sac development. Bisporic you can take the example of allium, and tetrasporic you can take the example of adoxa. But most of the angiosperms will follow monosporic type of embryo sac development, okay, which is also said as polygonum type of embryo sac. Okay, monosporic type of embryo sac development. Fine, right. So here you see, which is 7-celled 8-nucleated condition. So the most common type of embryo sac development is monosporic type of embryo sac development, which is also known as polygonum type embryo sac. So this has 7-celled 8-nucleated condition. It develops from single functional megaspore of the megaspore tetrad with the another 3 megaspores degenerated. This process is termed as monosporic development. So for your keen idea, I will give you a diagrammatic representation. See beta. So this is the mature embryo sac. How this mature embryo sac is formed? Okay. Listen first, you understand and then you go through. So here if you see, most of the angiosperms will be having what type of ovule? Most of the angiosperms will be having anatropous type of ovule, which is the inverted ovule. So here if you see the inverted ovule, that is anatropous type of ovule like this. This is the inverted ovule. Okay. At the center you have all the types of cells. So here it is the micropylar end, and this will be the chaliceal end. Close to the micropyle you will have funicle, which is the stalk of the ovule. So here if you see beta, all the cells which are present inside the ovule are called as what type of cells? Nucellus cells. Nucellus cells ploidy will be diploid only, yes or no? One of the nucellus cells at the micropylar region will be differentiated to megaspore mother cell. Okay, this is differentiated to megaspore mother cell. Fine, right? So this megaspore mother cell will first undergo meiotic division or reductional division, which will result in the formation of 4 haploid megaspores, okay, which is a linear tetrad. These 4 are haploid megaspores, of which the 3 megaspores which are towards the micropylar end will be degenerated and 1 megaspore towards the chaliceal end will be functional. This one functional megaspore will undergo 3 sequential mitotic divisions. This will undergo 3 sequential mitotic divisions. First mitotic division will result in formation of 2 cells. Second mitotic division will result in again formation of 2 cells. Total 4 cells, right, at the end of second mitotic division. Now third mitotic division will give out 8 cells. But here the thing is, the cells are not formed. What happens is, the embryo sac, in the embryo sac, after 3 mitotic divisions, after 3 sequential mitotic divisions, 8 nucleate condition is seen, because karyokinesis is not followed by cytokinesis at the end of each mitotic division. Okay, at the end of each mitotic division, karyokinesis is not followed by cytokinesis. So here after 3 sequential mitotic divisions only you can see cytokinesis formation, of which the 1 cell from the chaliceal end will come to the center and 1 cell from the micropylar end will come to the center, forming a central cell or polar nuclei. Okay, and 3 cells which are at the micropylar end will form egg apparatus, of which 2 will be synergids. Synergids will have filiform apparatus. This filiform apparatus will help in guiding the pollen tube towards the embryo sac. So here synergids and egg cell together called as what beta? Egg apparatus. Egg apparatus. Fine, right? Synergids and filiform synergids and egg cell together is known as egg apparatus, and at the center you can see central cell or polar nuclei and the 3 antipodal cells which are at the chaliceal end. Once again quickly I will recall, you just listen. This is very important both for NEET and EAP set also. This concept you need not to mention in your answer, but then to explain the embryo sac you have to know how embryo sac is formed inside the female reproductive structure. So in gynecium you have ovary. Ovary will consist of ovules. What type of ovule you will see in most of the angiosperm? Inverted ovule. So here is the micropylar end, this is the chaliceal end. Ovule will be composed of what type of cells? Nucellus cells. So this nucellus cells, here one of the nucellus cell at the micropylar end will be differentiated into megaspore mother cell. Megaspore mother cell, after undergoing reductional division, 3 megaspores, 4 haploid megaspores will be formed in a linear fashion, of which 3 megaspores towards the micropylar end will be degenerated and 1 will be functional megaspore at the chaliceal end. This functional megaspore will be undergoing 3 sequential mitotic divisions. In these 3 sequential mitotic divisions only karyokinesis will occur, cytokinesis will not occur. So here if you see, at the end of 3 mitotic divisions, 8 nucleated and 7 celled condition is formed, wherein 1 nucleus from the chaliceal end and 1 nucleus from the micropylar end will come at the center, forming central cell, and this is the polar nucleus, and 3 antipodal cell at chaliceal end, three cells which are towards the micropylar end are known as what? Synergids and egg cell together called as egg apparatus. This is the description on how embryo sac is formed. So, embryo sac is at last it is 7-celled 8 nucleated condition beta. Till here it is clear, no? Till here it is clear, right? Yeah. Next you see, so first you have to describe on what type of, most of the angiosperm will form what type of embryo sac and what will be the functional megaspore and how the embryo sac is formed. This is what you have to explain. Then give a detailed diagram of embryo sac, then start on describing the diagram of the embryo sac. Here if you see egg apparatus at the micropylar end, at the micropylar end you will have egg apparatus which is composed of 3 cells. 3 cells is 1 egg cell and 2 synergids. It is composed of 3 cells, 1 egg cell and 2 synergids. So, here you see egg cell is the central large with functional which functions as what? Female gamete. Remember, egg is the female gamete, embryo sac is the female gametophyte which is bearing the egg cell. Synergids are smaller, located on the either sides of the egg cell. So, if this is the egg cell, synergids are located on the either sides of the egg cell. Clear, right? They have filiform apparatus, cell wall thickening at the micropylar end. It has thickened cell wall at the micropylar end which facilitates the absorption of nutrients, and this helps in guiding what? Pollen tube. This will help in guiding of pollen tube into the embryo sac. Next is what? Central cell. Central cell is the largest cell of the embryo sac and this contains 2 polar nuclei which will later fuse. Okay, 2 polar nuclei which are at the center, they will fuse to form what? Secondary nucleus which is diploid. This forms secondary nucleus which is diploid. During fertilization, this nucleus will fuse with what? This secondary nucleus will fuse with male gamete and this forms what? Triploid primary endospermic nucleus. This forms triploid primary endospermic nucleus and antipodals at the chalazal end. Antipodals at the chalazal end, this consists of 3 small cells at the chalazal end. Okay, this will consist of antipodals which are at the chalazal end. This consists of 3 small cells. They are ephemeral, they are short lived and usually they will degenerate after or before the fertilization. This will function as vegetative or supporting cells. Okay, this will function as what ma? Vegetative or supporting cells. Sometimes this will help in nutrient transfer as well. So, the role of synergids, they will provide nutrition to the female gametophyte by absorbing nutrients from the nucellus. So, from the nucellus cells only the female gametophyte will be absorbing the nutrients. The filiform apparatus secretes chemical signals that will attract or guide the pollen tube towards the synergids for fertilization. This is the keen description of what? Central cell, of antipodals and synergids. After clear cut description, you have to mention a flow chart. If you give this flow chart, it will be more attractive in your exam. Present this flow chart, this will be more attractive in your exam. Okay. So, functional megaspore, here it is monosporic development. Functional megaspore, monosporic development, which is 7-celled 8 nucleated condition. Micropylar end and egg apparatus, they will have 3 cells, 1 egg cell and 2 synergids. Fine, right? Next, chalazal end, they will have 3 small cells, those are antipodals. Next, these synergids, and central cell, which is the largest cell at the center, they will have 2 polar nuclei. This is the secondary nucleus. This fuses with male gamete. This polar nuclei will fuse with male gamete, forming what? Endosperm. Clear, right? Yeah. Next, you see mnemonic to remember the angiospermic embryo sac. This mnemonic you should not write it anywhere in your exam, I am telling you. This mnemonic is to remember the answer in a sequential manner. Most of the students will not require any mnemonic to remember this answer, but few of the students, if you require at all, you can remember every cool ant. E for egg apparatus, C central cells, A is antipodals. For function and use, nourish, they will NG. okay, that is nourish embryo sac and guide the pollen tube via filiform apparatus. This thing you can remember, but most of the students will not get any requirement of mnemonic to remember this answer, because this is very simple and basic to remember about embryo sac, okay. And then if you see, draw the diagram of microsporangium and label its wall layers. Write briefly about the wall layers. We have to write about the wall layers more briefly. So here if you see about wall layers, the question is given in May 24, June 23, March 2023, October 2021, May 2018, March 2018 and May 2014. In every consecutive years you can see, okay, to describe the structure of microsporangium. So here when you are describing the structure of microsporangium, what are the layers you have to highlight? That is anther wall layers. 4 anther wall layers you have to highlight, okay. So here if you see answer, first thing is you have to describe about the microsporangium. A typical anther is 4-sided structure consisting of 4 microsporangia located at the corners, 2 in each lobe, 2 microsporangia which are present in each lobe. So here if you see, the microsporangia will extend longitudinally throughout the anther and are packed with what? Pollen grains. That is why you will call each lobe as what? Pollen sacs. After pollen grains get matured, you will call out each lobe as what? Pollen sacs. So here you see ma, this is the diagram. First I will explain you this complete bilobed anther structure, which is very important, not just for your IPE exam, it is also important for your NEET and EAP set. So here you see, first you have to describe on the wall layers. So anther is having how many wall layers? 4 wall layers. First is what? Epidermis. First is epidermis. Second is endothecium. Third will be middle layers. Fourth is tapetum. If you have to see the ploidy, epidermis, endothecium and middle layers, 3 ploidy is diploid only. Tapetum ploidy will be polyploid. This is the innermost wall layer of the anther. This polyploid anther, they will give what? Nourishment to the sporogenous tissue which is present at the center. Okay, tapetum will give nourishment to sporogenous tissue which is present at the center. At the center you will have connective tissue. It will connect the sporogenous tissues which are present in 2 lobes. Okay, now in the answer you have to systematically describe about each wall layer of the anther. So look into it clearly, ma. Look into it clearly. Microsporangial wall. Each microsporangium has 4 distinct wall layers. For example, first is epidermis. You have to give out the structural description, then functional description of each layer. Okay. First you look at the structural description, epidermis. This is the outermost single celled continuous layer of the anther wall. Here cells are small and thick walled. Okay. Cells are small and thick walled. If you see the functions, they will provide what? Protection and modified thin walled cells at the junction between pollen sacs. They will form stomium which will help in dehiscence. So epidermis, modified thin cells of epidermis, they will form the junctions of the pollen sacs here. So the epidermal wall layer at the junctions, they will get like thin walled cells, they will get modified to what? Stomium, and this stomium region will help in the dehiscence of the pollen grains at their maturity. Okay, very important, you have to highlight the word stomium when you are writing the answer. Now endothecium, second is what? Endothecium, middle layers, and then is tapetum. So here you see endothecium, this will lie below the epidermis and cells will develop cellulosic thickenings on the tangential walls. Okay, this will give out what? Cellulosic thickenings on the tangential walls. At maturity, these cells will lose water and they will contract. Okay, this will create a tension that helps in anther dehiscence and pollen release. They will help in the anther dehiscence and pollen release. Middle layers you look into, ma, middle layers, they will lie beneath the endothecium. Middle layers are present beneath the endothecium. Usually they are 2 to 3 layers of flattened thin walled cells. these layers, they will degenerate in the later stages of development and they will have minor role in the anther functioning. Next is tapetum. In tapetum you see this is the innermost nutritive tissue, this is surrounding the sporogenous tissue. Sporogenous tissue, which is 2 sporogenous tissues are present in the each lobe of anther. And cells are large, dense, they are often multinucleated condition with abundant cytoplasm. Tapetum is having abundant cytoplasm. What are the major functions of the tapetum over here? They will provide nutrition and nourishment to the microspores, of course that is there. They will secrete enzymes, hormones essential for pollen exine wall. Exine wall formation means the tapetum itself is producing sporopollenin. Okay, tapetum is helpful in the formation of sporopollenin. Fine, right? They will play major role in pollen development and maturation. Okay, they will play major role in pollen development as well as maturation. So, to mostly mnemonics are not required to remember this answer as well, but for few students I am giving out. Every end made tough, like epidermis, endothecium, middle layers and tapetum as such. So, how you have to go with this answer beta? First describe the microsporangium, then draw out or give out the diagram of microsporangium with the labelings, then start on giving the explanation about each layer of anther wall, and at the center you also have sporogenous tissue, no? That also you have to explain in detail, right? Clear, right ma? Now you see the last question, describe the process of fertilization. Just a second. Yeah. Two more questions from this chapter. First is describe the process of fertilization, which is asked in quite a few years, May 2022, October 2021 and March 2015. 3 consecutive years you got a question on fertilization of angiosperms. How this will happen? First you have to start on explaining pollen pistil interactions, then you have to explain about double fertilization. So, here you look into fertilization is what? This is a process of fusion of male and female gametes in angiosperms. So, this will result in formation of zygote and endosperm. Zygote whose ploid is what? Diploid, and endosperm ploid is triploid. It takes place inside the embryo sac of the ovule. Here you see pollen pistil interaction. The pollination will bring the pollen grains into the stigma. Stigma will test for the compatibility of the pollen. If you see self incompatibility, that pollen grains of the same flower will not be accepted by the stigma. As such, many pollen grains will land on stigma, but stigma will not accept until the compatible pollen grain will come and land over the stigma. So, here you see if pollen is compatible, the stigma accepts it and allows for germination. The stigma will accept it and allow for germination. If pollen is incompatible, the stigma will reject it and this will prevent the germination. In compatible pollination, what happens? In compatible pollination, pollen grain germinates on the stigma, pollen tube will grow through one of the germ pores and the tube penetrates the stigma and style, carrying the 2 male gametes. This will carry 2 male gametes towards the ovule. Okay, so here we go with the diagram. Okay, so in this longitudinal section you have a pollen tube with antipodal, synergids and egg cell, embryo sac is given, ovule is given and complete pistil is drawn. Okay, this diagram you have to practice in a more detailed way, because diagram first should be impressive. Okay, you have to give an impressive diagram with a neat labeling and then start on describing the answer in a systematic way. Okay, so this is about pollen pistil interaction. Here you see the stages of fertilization. First is the entry of pollen tube. Entry of pollen tube can be through 3 methods. One is called as porogamy, second is mesogamy and third is chalezogamy. So, entry of pollen tube, if it is from micropylar end like this is called as porogamy. Okay, most of the angiosperms it is porogamy only. If the pollen tube is coming from the integuments, if pollen tube is arising from the integuments, this is known as mesogamy. Mesogamy is mostly observed in cucurbids. And if pollen tube is arising from the chalazial end, it is known as chalazogamy. Best example is Casuarina. Fine, right? So here we have porogamy, mesogamy and chalazogamy. This is for what? Pollen tube entry. So the very first point that you will be describing when you are starting about the stages of fertilization is entry of pollen tube. So the pollen tube enters the ovule through micropyle in most of the angiosperms. It penetrates into the synergids, wherein filiform apparatus will be degenerated. One of the synergid will be degenerated because of the entry of pollen tube. Then release of the male gametes into the embryo sac. Then starts the concept of double fertilization, wherein one of the male gamete will fuse with egg cell forming a diploid zygote. Another male gamete will be fusing with the central cell forming out primary endospermic nucleus. This primary endospermic nucleus will develop into triploid endosperm which nourishes the developing embryo. Next is entry of pollen tube into the ovule. There only you can write it, or here also you can explain, that is your comfort beta. That is porogamy, chalazogamy and mesogamy. Fine, right? Next, stages in fertilization. First is discharge of male gametes, next will be syngamy, then triple fusion. So here you see syngamy or triple fusion we will call it because the second male gamete will fuse with two polar nuclei in the central cell. This forms a triploid primary endospermic nucleus. Double fertilization is unique to angiosperms. The occurrence of both syngamy and triple fusion in the same embryo sac is seen. So first time double fertilization is reported by Navastin in the year 1898. Okay, this first double fertilization was first reported by Navastin in the year 1898 in Lilium and Fritillaria. Okay, this is reported by what? Lilium and Fritillaria. And these are the diagrams of pollen tube entry and how synergids are getting disrupted and how male gametes are being released. Now at the last you have to give out the significance of double fertilization. Just observe, monitor the diagrams, ma. Just observe and monitor the diagrams. I will be back in 1 minute. Here we go with significance of double fertilization. This ensures the zygote formation. Zygote formation was ploidy is diploid, and then this will produce triploid endosperm, a special nutritive tissue for embryo development. This will establish a link between sexual reproduction and seed formation, making the process highly efficient. This will provide an evolutionary advantage. Nourishment of embryo is guaranteed only after fertilization occurs. So this is the significance of double fertilization. Just 3 to 4 points if you give in significance, that is enough. Next, a mnemonic to remember it in sequence. Okay, pretty cats made sweet tea daily. Okay, pollen pistil compatibility or pollen pistil interaction here. So this answer may require the mnemonic support, okay, to most of the students. So you look into it keenly. First, P, pollen pistil interaction you have to mention. Second, followed by compatibility of stigma. Third, male gametes are released by the pollen tube. Fourth, syngamy, that is fusion between male gamete and egg cell. Last, triple fusion, and give out double fertilization sequence at the end. Okay, so this is how you have to present this question. It is your comfort, like where you are giving out the diagrams, but highly most of the time you choose diagram based questions only beta, because diagram based questions more marks will be awarded for that. by diagram and its systematic explanation. Okay, so this is about fertilization, and it will be very impressive if you produce the flow chart for fertilization in angiosperms. Flow chart, you maximum you produce out the flow chart for each long answer. It will be very impressive. So see, pollination followed by pollen and pistil interaction, next compatibility, whether it is compatible or incompatible, next pollen tube entry, it can be through porogamy, chalosogamy and mesogamy. Here you can write out the examples also, like Oinothera, Casuarina and cucurbits. Next, discharge of male gametes, and then double fertilization, which is syngamy and triple fusion, and then post fertilization products like what are formed at the end of fertilization. At the end of fertilization, you can see zygote will be turned to embryo, will be developed to embryo. Primary endospermic nucleus will be matured to endosperm. Ovule turns to seed. Ovary will turn to fruit. Okay, these are the post fertilization products. Next, write a brief account on agents of pollination. Okay, agents of pollination, you see, first definition, pollination is what here? Transfer of pollen grains from anther to stigma of the flower. Okay, agents of pollination can be biotic or abiotic. You can see this question is also important, where it is asked in 2022 and March 2017. So here you see agents of pollination, it can be biotic or abiotic. First, in the types of pollinating agents, we will go through abiotic pollinating agents. So abiotic pollinating agents includes majorly wind pollination and water pollination. Wind pollination is addressed as anemophily, water pollination is addressed as hydrophily. This hydrophily can be epihydrophily and hypohydrophily. Epihydrophily and hypohydrophily. So here epihydrophily, hypohydrophily, these are the abiotic pollinating agents that we are going to discuss. First, anemophily, wind pollination, where light, dry, abundant pollen grains will be carried out by wind. Flowers are usually small, inconspicuous and without any nectar. Examples you can see maize, wheat, grass. These are the examples for anemophily. Next is hydrophily. Pollen grains will float and they are carried by what? Water currents. Pollen grains will float and pollen grains are carried out by water currents. They are found mostly in the aquatic plants. Pollen grains are found mostly in the aquatic plants. For example, here you can see Vallisneria and Hydrilla. Okay, Vallisneria and Hydrilla. Okay, ma. Examples include Vallisneria and Hydrilla. Clear, right? So next, if you observe, yeah, this is the pollination by water in Vallisneria. Biotic agents. Biotic agents include insects, birds. Insects, entomophily. Most common agents of biotic pollinating agents, most common they are insects like bees, butterflies, moth, beetle and flies. Flowers are colorful here, they are scented and they produce nectar to attract insects. Examples include hibiscus and sunflower. Next, birds, ornithophily. Birds like hummingbirds and sunbirds, they will act as pollinators. Flowers are large, tubular, brightly colored and they will produce abundant nectar here. Examples include begonia and bombax. Okay, begonia and bombax. These are the examples for bird pollination. Next, chiropterophily. Here bats will visit the flowers at night. Nocturnal pollinators they are, and flowers are large, dull colored, they will emit strong odor. Examples here include Boinia and Kygelia. Other animals include what? Small animals, squirrels, lemules, snails. They are also occasionally they will act as pollinators. So the agents of pollinators, complete mnemonic, here you can see. Pretty wise woman inspire bright beautiful offspring. Here you see, first P is pollination definition you have to mention. W, wind. W is wind. Abiotic anemophily here. water pollination, hydrophily, insects, entomophily, birds, ornithophily, bats, chirapterophily, and other animals like squirrels, lemurs and snails. This is the best mnemonic to be addressing this question. Fine, right ma? So, what are the 4 questions that we have discussed today from sexual reproduction of flowering plants? First is about the structure of embryo sac, second pollen pistil interactions, third will be microsporangium, and 4 is pollinating agents. Okay. So, first is embryo sac, pollen pistil interactions, second is third is pollinating agents, and fourth is about embryo sac structure. Embryo sac structure, pollen pistil interaction, double fertilization, anther microsporangium structural description, and then lastly is pollinating agents. These are the 4 concepts to be highlighted from this chapter. Once again I am listing out ma, just see. First is double fertilization, second question is about embryo sac, next structural description of microsporangium, and fourth question will be about pollinating agents. Fine, right. This is about pollinating agents. So, here these concepts from this chapter are very important. I am telling you, not just for IPE but also for NEET and MCET. Okay, monitor and learn them accordingly. Fine, right ma? So, that is all for today's session, and with today's session we have finished out discussing all the long answer type of questions from morphology, anatomy and sexual reproduction of flowering plants from first year BIPC. From our coming class we are going to discuss the questions on short answer type of questions. Okay, short answer type of questions from IPE first year BIPC. Okay. So, stay tuned and keep learning. These revision classes will be helpful to you a lot. Okay. So, stay tuned and learning Vita. Thank you. That is all for today.